learners, this is Binduprava Sahu. Welcome to SNEB'S Biology. In the previous class, we have discussed about how to draw a typical plant cell. In this class, I will tell you some tricks and tips how to draw a typical animal cell. Okay, so let's first start with the trick. To remember the various components of a typical animal cell, you need to remember a sentence again. So, what is the sentence? What is the new sentence? The new sentence is Mrs. Rath noticed Pia in the college. Okay? Mrs. Rath noticed Pia in the college. Now, remember the block letters. MRS, R, N, P and CLG. Okay. CLG again is the short form of college. So, let's write the initials first. Or what are the letters written in the block letter? M, R, S, R, N, P and then C, L and G. Let's not write it here. C, L and G. So, M for mitochondria. Okay. Then R for ribosome. Then S for smooth endoplasmic reticulum. Okay. Then R for rough endoplasmic reticulum. Then N for nucleus and then P for plasma membrane. Then C for centriole. Remember, centriole is the characteristic feature of a typical animal cell. It is not present in case of plant cell. Okay. Then lysosome. Then Golgi complex. Okay. So what are the structures? Mitochondria, ribosome, smooth endoplasmic reticulum, rough endoplasmic reticulum, nucleus, plasma membrane, centriole, lysosome and Golgi complex. MRS, RNP and CLG. Okay, remember this sentence? Mrs. Rath noticed Pia in the college. Okay, clear? Now, let's learn how to draw the typical animal cell using these 9 components. Okay? Now, let's start drawing an animal cell. Okay? So, first, let's write the various components in their short form so that we will not miss out anything. Okay. So, M, R, S. Okay. Then, R, N, P. Then, C, L and G. Okay. So, what is the outer wall of the animal cell? The outer wall of the animal cell is the plasma membrane. Remember that there is no presence of cell wall in case of animal cell. Okay, the outer wall will be the plasma membrane. So let's draw the plasma membrane. Okay, this is the plasma membrane. Okay, so in the plasma membrane there are various structures are present. Typical structures called as the microvilli. Okay, microvilli. So what are the microvilli? The microvilli are the membrane outgrowth. Okay, which help in increasing the surface area of absorption. Okay, so they are finger-like projections. So let's draw. And these are the microvillar structure. Okay, clear? So in the plasma there is the plasma membrane. In the plasma membrane there is microvilli. Okay, which will increase the surface, which at the finger like projections, increasing the surface area for absorption. Okay, so in the membrane, what is the membrane? The plasma membrane. This structure is complete. Now let's come to the nucleus part. Okay, so let's draw the outline of the nucleus. This is the nucleus. Now we will draw the membranes. Okay, nuclear membranes. This is the nuclear membrane, and in the nuclear membrane there is presence of nuclear pore. So in the inside the nucleus there is the nucleolus. Okay, let's divide it like this. There is nucleolus, and there are many chromatin networks. Okay, so what are the chromatin networks? There are two types of chromatin network found inside the nucleus, euchromatin and the heterochromatin. We will draw both. So these are the euchromatin and these are the heterochromatin. Okay, yeah, euchromatin as well as your heterochromatin. So the nucleus is complete. Okay, we have drawn one open three-dimensional structure of the nucleus. Okay, so surrounding the nucleus is complete. Now surrounding the nucleus there is endoplasmic reticulum. So how many types of endoplasmic reticulum are there? There are two types of endoplasmic reticulum: smooth endoplasmic reticulum and rough endoplasmic reticulum. So what is rough endoplasmic reticulum? The endoplasmic reticulum which contains the ribosomes. Ribosomes are associated within the membrane of the endoplasmic reticulum. That kind of endoplasmic reticulum is known as your rough endoplasmic reticulum. And what is smooth endoplasmic reticulum? The endoplasmic reticulum which is devoid of any ribosome. Okay, so let's draw. One, there is endoplasmic reticulum. H, we will draw many H, connected H. Okay. Then let's draw here. Okay, now here okay. These are the endoplasmic reticulum surrounding the nucleus. Some are also generated from the nucleus. Okay. Okay. So if we are drawing rough endoplasmic reticulum, then we will also draw some ribosomes. Okay. So these are the ribosomes. Okay. Then let's draw some smooth endoplasmic reticulum which is devoid of ribosome. Okay. So These are some smooth endoplasmic reticulum which do not contain the ribosome. Okay. So smooth endoplasmic reticulum is complete and rough endoplasmic reticulum is complete. Now let's come to ribosomes. So besides the smooth and rough endoplasmic reticulum, there are some ribosomes which lie in a scattered manner inside the cytoplasm. Okay, so let's draw them. So these are some free ribosomes. Okay, these are some free ribosomes. Now let's come to the mitochondria. Okay, this is the mitochondria. So let's draw. There are some beans like structure which have inner projected compartments. Okay. The number of mitochondria in a cell depends upon the metabolic activity of the cell. More the cell is the metabolic active, there are more numbers of mitochondria. Okay, let's draw only 3. So this is the mitochondria. Okay, now let's come to the Golgi body, G. Okay, so draw the Golgi body. They are more like some tubular and dumbbell shaped structure. Which are present in the cell. Okay, and there are some vesicles which come out of the Golgi body. Okay, clear? Now come to lysosome. So what are lysosomes? These are small rounded structures present in the cell. It is also present in the animal cell. Okay, plant cell. We have drawn that. Okay. Now this is the last one is the centriole. Okay, so let's draw the centriole. The centriole are bundle like structures. Stick bundle, remember in your school you draw it like this, right? So this is what? The centriole. Okay, this is the centriole. Okay. So we have completed all the structures. Besides these structures, some additional structures are also present in a cell. Okay, in the animal cell, like the microfilament and the microtubules. Okay, so let's draw them. These are some microtubules. Okay. Some microtubules, and let's draw some microfilaments. Yes, these are some microfilaments. Okay, then come to the Golgi vesicles. Okay, the Golgi vesicles. So they are like this. These are the Golgi vesicles. Okay, and also there are some secretory vesicles. Okay, so let's draw them like this. These are some secretory vesicles. Okay, these are the secretory vesicles. Okay, this much is the structure. So now let's label them. All these extra structures you may draw as per your preference. Okay, if you remember them, then draw, otherwise leave them. Okay, this much is necessary. The 9 structures which I have told you, that much is necessary. At least you have to draw that much structure if it comes in your examination, to draw the diagram of a typical animal cell. Okay, clear? So now let's label them. So what is this? This is microvilli. Okay, this is microvilli. This is plasma membrane. Okay, what is this? This is plasma membrane, the outer wall of the animal cell. Okay, this is secretory vesicle. Then this is Golgi vesicle. Okay, then this is lysosome. Okay, then what is this? What is this structure? This structure is the Golgi body. Golgi body or the Golgi complex. Okay, now what are these? These are the microtubules. Okay, clear? Then what is this? This is the centriole. This is the centriole. Okay, now what are these? These are the smooth endoplasmic reticulum, SER. And what are these? These are the rough endoplasmic reticulum. Okay, clear? Now what is this? This is the nucleus. This is the nucleus, and this is the nuclear membrane. Okay, we have cut off the nucleus. Then this is the nuclear membrane. This is the nuclear membrane. And what is this? This is the nucleolus. Okay, and there are various chromatins present inside the nucleus. What are those? Those are the chromatin network. Chromatin network. And in the chromatin network, there is also presence of euchromatin and heterochromatin. Okay, heterochromatin. Here, now what is this? This is mitochondria. This is mitochondria. And remember the nuclear pore. What is nuclear pore? In between the two nuclear membrane. Okay, in between the nuclear membrane there are some gap. That is called as the nuclear pore. So let's write it here. Nuclear pore. Okay, clear? Now, then what are these? These are the microfilaments. Okay, then there are some ribosomes which lie scattered in the in the cell. These are the ribosomes. What kind of ribosomes are these? These are free ribosomes. Clear? And the cell matrix is called as the cytoplasm. Okay, clear? So this is the labeling of a typical animal cell. What is this? It is an animal cell, ultrastructure of an animal cell. Okay, understood? So this much for today's class. In this class we have discussed how to draw a typical animal cell, and remember the trick, the sentence, that is Mrs. Rath noticed Pia in the college. Okay, clear? तो ये जो हमारा जो रजिस्ट्रेशन स्टार्ट हुआ है वो 31 जुलाई को खत्म होने वाला है. सो जल्दी जल्दी रजिस्टर कर लीजिए अपने आप को. डू सब्सक्राइब एंड सपोर्ट अस शो योर लव बाय गिविंग इट अ थम्स अप, शेयर इट विथ योर फ्रेंड्स। ओके, हमारे वीडियोस आप लोगों के लर्निंग के लिए है। आपको बहुत सारे ट्रिक्स एंड टिप्स ऐसे बताए जाएंगे, वेरियस सिलेबस आपके कवर अप होंगे। ओके, सो डू लाइक आवर वीडियोस। थैंक यू।